This chapter is about histology, and histology is the study of tissues, so this is the foundation for all the chapters that we will be studying next. So this is an important chapter for you. Please study this chapter, and everything in this chapter will be fair game for the test. Every single part of this chapter is important. This chapter will deal a lot with the principle of complementarity of form and structure, and just remember that this means everything in the body makes sense, that form follows structure. So there are 4 tissues in the body: epithelial tissue, connective tissue, nervous tissue, and muscle. And this is just a link to a great video about the cell. Now the cell was in chapter 3, but since I didn't have this link for chapter 3, I just thought I would throw this in here. So if you want to copy this link down, go to this. This link will actually take you to the Harvard Multimedia website, and you'll have to find the link on the cell itself. I showed this in class, so it's not obvious where to go to, so just call me and I'll tell you where to go to on the website if you want to see the video that I showed in class, if you're an in-class student. So we're going to start with epithelial tissue, and the jobs of epithelial tissue are going to be down here. But first, in general, epithelial covers everything in the body, or it lines everything in the body. So that's how you're going to be able to identify it if you're looking through something, if you're looking through a microscope. Is it covering the outside of an organ, or is it lining the inside of an organ? And then we're going to go over glandular epithelium later on in this lecture. But our roles of our epithelial tissue will be boundaries, like for instance, they create boundaries between organs, or for instance, our skin creates a boundary between the air and our body, and they provide protection from abrasion, protection from pathogens on our skin surface, etc. Absorption and secretion are big functions of epithelial tissue, to absorb nutrients, for instance, across the cell in the gut, and allow those nutrients to be absorbed into the blood. For secretion, secretion is when a cell secretes a product that it makes right into the lumen of whatever that epithelial tissue is lining. So it might secrete a hormone that it makes, it might secrete mucus right into the lining. Filtration is when a solvent in a solute is being pressed by pressure through a membrane. So filtration often occurs through epithelial tissue. It's going to occur through the thinner layers of our epithelial tissues. Excretion is when we get rid of unnecessary products, and that happens often through our epithelial tissue. And sensory reception. Sensory reception is detecting something that is occurring, say on our skin. If a fly lands on your skin, you're going to detect that because you have sensory receptors in the skin. However, sensory reception is actually occurring because of our nervous system, but the nervous system is housed in our epithelial tissues. So epithelial tissue is not actually receiving that information. The sensory receptors are just housed in the epithelial tissue. So when it comes to the epithelial characteristics, these are our epithelial characteristics, and one big characteristic of epithelial tissue is the fact that it exhibits polarity. That means one surface, the top surface, which is the apical surface, does not look like the bottom surface, which is the basal surface. That's polarity. Sometimes we're just one cell layer thick, and if that's the case, the top doesn't look like the bottom. And if we're many cell layers thick, the top surface doesn't look like the basal surface. Epithelial tissue also has special contacts, special cell-to-cell contacts like tight junctions. We went over that in chapter 3. Tight junctions prevent leakage between cells. And desmosomes. Desmosomes are special cell-to-cell contacts that prevent cells from being ripped apart from one another. They're also called adhering junctions. Epithelial tissue is always supported by connective tissue. That means connective tissue will always be underneath the epithelial tissue, because epithelial tissue does not have its own blood supply. Avascular means without blood vessels. That's what avascular means. And so the epithelial tissue has to depend on the underlying connective tissue for its oxygen and its nutrients. But just because it doesn't have blood doesn't mean it doesn't have blood vessels. epithelial tissue has a very good supply of nerves. And I like to remember this because if you get a paper cut, you don't bleed, but you do feel it. So your epithelial tissue, for instance, is covering the outside of your body. It's your skin. So remember, paper cuts. You don't bleed, but you feel it. Now, also epithelial tissue has the best regenerative capability of all the four tissue types of the epithelial, muscle, nervous, and connective. Epithelial will be have the best regenerative capability. If you damage it, it regenerates very well through mitosis. Now, to classify our epithelial tissues, we have two ways to classify it. First, we're going to classify according to the number of layers that are present. And the second way, we're going to classify according to the cell shape. So if we have simple epithelium, we're only going to have one layer of epithelial cells. If we have stratified epithelium, we're going to have more than one layers. The simple epithelia are found in places where we're going to be absorbing substances or secreting substances, or we have filtration that's occurring. And I should have put diffusion here as well, because diffusion also occurs. So if you want to add that into your notes, we have absorption, filtration, diffusion, or secretion occurring in places where we have simple epithelial tissues. In stratified, where we find stratified epithelial tissue, we find those in places where lots of abrasion is going to occur. Our second type of classification is according to cell shape, and we're going to have flat cells, very flat cells. Those are called squamous cells or squamous cells. We have cube-shaped cells. Those are cuboidal cells. And then we have column-shaped cells called columnar cells. Now this is going back to this polarity issue. We have the top of the cell looks different than the bottom of the cell. This is a simple epithelium because it's one cell layer thick. This is the apical surface up here, and this is the basal surface down here. This is the stratified epithelium because it has many cell layers, and this is the apical surface up here, and this is the basal surface down here. Now the apical surface is going to be the free surface because up here it's free. Up here it's free. The basal surface is going to be in contact with what we call the basement membrane. Down here is the basement membrane. Here's the basement membrane. And we call it the basement membrane, but the basement membrane is going to be composed of two things, the basal lamina and the reticular lamina. The basal lamina is a bunch of sticky proteins called proteoglycans. And so we'd say this top layer up here might be proteoglycans and some collagen fibers. And those proteoglycans and collagen fibers are secreted by these cells up here in the epithelial layer. And it's a sticky like glue to stick this epithelium to the underlying connective tissue. This lower part of the basement membrane, called the reticular lamina, is also composed of proteoglycans and collagen fibers. And those little tiny collagen fibers are also called reticular fibers. That's why we get the name reticular lamina. But the reticular layer is secreted by this underlying connective tissue that's down here. So part of the basement membrane is made by this epithelial tissue, and part of the basement membrane is made by the underlying connective tissue down here. Now when we look at these cells from the side, here are three cell shapes. We have squamous, which is a sort of a flat cell with a flattened nucleus in it. The cuboidal cell shape is a cube-like cell, and it has a round nucleus. And the columnar cells are column-shaped, and they have a more oval-like nucleus. From the side, they all look different. From the top, if we look from the top, especially underneath the microscope, they all have this six-sided cell shape. They kind of look like a fried egg from the top. They all look the same from the top. Sometimes these squamous cells look like a line, a straight line, like this line like here, with a little black mark on it for the nucleus. They don't look like this at all. Like this right here. That looks like a fried egg right there. That's a squamous cell right here. You can't tell where one begins and the other one ends, but there's the nucleus of that squamous cell. And this is what a squamous cell looks like from the top. It looks like a fried egg. Here's from the top with its nucleus, and here's from the side with its nucleus. So they look totally different from the side and from the top. But here's our first epithelial tissue, simple squamous epithelium, and this is what it looks like in the lungs. So down here in this round picture, it's showing you where we're taking the sample, where they've taken a little red picture. It's actually could be anywhere out here from these regions out here in these microscopic regions of the lungs. This is the alveoli of the lungs out here, the air sacs. All of this up here, every bit of this that I'm lining with my mouse, this is all simple squamous epithelium. These are little simple squamous cells basically holding on to one another. And it's a very thin layer. And these are the air sacs of the lungs. This is where the air would be filling up in here. All these little things that you see in here, they could be other types of cells because there are other types of cells in the lungs, but they look to me to be simple squamous cells that have been sloughed off in preparation of this slide. So you can ignore all these cells right in here. They're just an artifact of the slide. These are the air sacs. So oxygen is going to be diffusing. Now oxygen usually doesn't diffuse from one air sac into the other, but really it's going to be diffusing into the capillaries. Pretend like this is a capillary right here. Capillaries are just as thin as the air sacs, and if we had oxygen in here, we want that oxygen to diffuse through the air sac into the capillary here and be carried to the rest of the body, to be diffused into the cells, to be sent to the cells, because our cells need that oxygen to make ATP, right? So the reason we have these simple squamous cells, only one cell layer thick, is we don't want a thick surface for that oxygen to have to diffuse through. So here we have complementarity of form and of structure and function. So the purpose of simple squamous epithelium is diffusion in this case. Now, if we were in the kidneys, the purpose of the simple squamous epithelium would be filtration. We would be pressing liquid through the walls of the simple squamous epithelium. We would be through the walls of the simple squamous epithelium. We would have water and solutes being forced through there and trying to cleanse our blood and forming urine at the same time. That's the process of filtration. So what I want you to do when you're going through here, you have to take a lab practical. You have to be able to identify what simple squamous epithelium looks like on a slide. You have to know its function, and you have to know where to find it in the body. So function, location, and how to identify it. Now this is what simple cuboidal looks like. Here's the artist drawing, and here's what it looks like if you were to fold that over into a circle. We've taken this out of the kidneys. That's where we find simple cuboidal in the body, in the kidneys, and its function is secretion and absorption. Here's simple cuboidal if we folded it over. Here's simple cuboidal right here, and here's the apical surface, and here's the basal surface right here. And so here's the basement membrane. So all this underlying stuff over here, all this stuff in here, is all connective tissue. Actually, this is a capillary and those are little red blood cells, but here's connective tissue and here's connective tissue. I see lots of capillaries in here. So all this would be connective tissue, because here's another ring of simple cuboidal cells. Here's another ring of simple cuboidal cells. Here's another one. Here's another one. These are all rings of simple cuboidal cells, and so here and here it's a little squished, but these would be open, hollow spaces through which urine would be flowing, or the precursor to urine would be flowing inside the kidneys. So this is all simple cuboidal here. And all this in this region would be connective tissue, the underlying supporting connective tissue. In this picture we have simple columnar epithelium, and we see in the body that it runs from the stomach all the way from the small intestines to the large intestines and through the rectum. And its job, just like simple cuboidal, is absorption and secretion. And here we have those simple columnar cells lined up in this region. Here we have our basement membrane down here, so this is all connective tissue down here. This is the basal region, and so this is the apical region up here. So here's our free surface. So these are our simple columnar cells down here, and we've taken a longitudinal section through a part of the small intestines here, a very small part of the small intestines called the villus. So this is a very small part here. So this is simple columnar epithelium, and here's another part of simple columnar epithelium. But now this part is upside down, so this is the apical region here, and here's the basal region here. So this is connective tissue on this region up here. Now this next slide is pseudostratified ciliated columnar epithelium, because here's the cilia, so they left the word ciliated out. You need to put that in. It's pseudostratified ciliated columnar epithelium, and we find this all along the respiratory tract, not just down the trachea, but we're going to find it a little farther down here. We're going to find it up also in your nasal passages. And these are goblet cells, and these goblet cells actually extend further down. You just can't see them. They look like goblets, and they're full of mucus. And that mucus is made of mucin. Let me get to this next page. This is what a goblet cell really looks like. This is in a different part of the body because this is not cilia. These are microvilli. But these goblet cells are full of little vesicles that will be secreted full of mucus. But right now, when it's in the cell, it's made of mucin. Mucin is the precursor to mucus. If you add mucin plus water, then you get mucus. So if we secrete this out and it gets into the water, and here we're in the digestive tract, this goblet cell's in the digestive tract. So if we secrete this out and there's water out here, we'll make mucus. We're going back to the respiratory tract. When this mucin is released out here, we'll have mucus out here on top of the cilia. It would be in here, and when we breathe in, all that dirty air would be trapped in our mucus, and the cilia would brush upward, upward, upward until we swallow it down into our stomach to make sure that only clean air gets down into our lungs. So the cilia is always beating upward, upward, upward. Now the reason this is called pseudostratified ciliated columnar epithelium, it's classified as a simple epithelium because all of the cells, the basal surface is down here and the apical surface is up here. It appears to be more than one layer of cells, but it really is only one layer of cells. All these nuclei here, looks like another row, but really we just have one layer of cells. Okay. Their purpose is secretion because they're secreting this mucus and beating it along, and they're really found all throughout the upper respiratory tract. So here's another picture of pseudostratified ciliated columnar epithelium. It's just a whole different look. See how confusing it looks? It looks like there are many layers, but this word means false layers, pseudostratified. It's really a simple epithelium. I just wanted you to get used to two different looks to that pseudostratified ciliated columnar epithelium. But if you see the cilia, that's a dead giveaway that it's this pseudostratified ciliated columnar epithelium. Now we're on to our first example of stratified epithelium. This is stratified squamous epithelium. We don't have any slides of stratified cuboidal or stratified columnar. They're not that abundant in the body. Stratified cuboidal is around glands, and stratified columnar is would be maybe in the male urethra, and that's about in some transitional locations. So this is the one that I want you to be familiar with because it's the most abundant, and it's the one we have a picture of and the one we will see in the lab. So stratified squamous is the one I want you to focus your attention on. It starts down here and it goes all the way up. This is stratified squamous. This is the non-keratinized type. In lab, I'll show you the keratinized type. The non-keratinized type has nuclei that goes all the way up to the top. The keratinized type has a bunch of waxy cells that look like they're going to flake off up here, and the nuclei do not go all the way to the top. The keratinized type is only found in the skin, and the non-keratinized type is found in the mouth, in the vagina, in the esophagus, in the anal canal. Those are some regions where we'd find the non-keratinized type. The reason we have to keratinize the skin is because the skin will dry out. And so we have to add a waterproofing protein called keratin to make sure our skin doesn't dry out. And so here's what our stratified squamous looks like. They're sort of cuboidal shaped cells here on the bottom, but as we move up, they're still a little kind of fluffy here, but as we move up, they flatten out considerably. So this is stratified squamous epithelium. And the stratified epithelia are stratified for a reason. Their job is protection, so we can slough off many layers and still be fine. Here's another stratified epithelium. It's called transitional epithelium. Now, we can slough off some of these layers and still be fine. That's what we do when we're a stratified epithelium. But we find this only in one place in the body. It's in the urinary system, and it goes from the ureters here. These are called our ureters, and it's lining the inside of the bladder and just in a small part of the urethra, because that's one of the other places we find this stratified squamous epithelium. I didn't list it down here, but in part of the urethra we find the stratified squamous epithelium. But here we go back to our transitional epithelium. It's called transitional because sometimes it looks like this with these big dome-like cells at the surface. They're very dome-shaped, and sometimes it looks like they have two nuclei in them. That's what I always look for. But it transitions between looking like this when there's no urine in the bladder to looking like this, thin and stretched out when there is urine in the bladder. So transitional looks like this sometimes, and it looks flattened out and stratified, like stratified squamous when it's stretched out and full of urine. So the function of transitional is to stretch out and accommodate a distension of the bladder. Now, there's glandular epithelium, and it's not classified like normal epithelium. We classify whether it has one cell or it's multicellular. And then we also classify it, is it endocrine or is it exocrine? Now, endocrine epithelium is we're going to cover it next semester. And an endocrine epithelium, glandular epithelium, it's part of the endocrine system. And basically it's this. If you're an endocrine gland, you do not have a duct and you secrete hormones. If you're an exocrine gland, you have a duct and you secrete non-hormone products. And examples of exocrine glands would be salivary glands, mammary glands, lacrimal glands. Now the pancreas is an example of an endocrine gland because it does secrete hormones, and it's also an exocrine gland because it does secrete some exocrine products like its digestive enzymes. But you're an exocrine gland only if you secrete your product out through a duct. And you can secrete that to the outside of the body or to the inside of the body. Another example of an exocrine gland would be your sebaceous glands. And so we've got lots of classifications going on here. You're either unicellular or multicellular, you're endocrine or you're exocrine. We're going to focus on our exocrine glands. Now, also another way to classify the glandular epithelium is, are you merocrine or are you holocrine? If you're a merocrine gland, you secrete by exocytosis, and most all of the exocrine glands secrete by exocytosis. The one exception, is the sebaceous glands. Sebaceous glands are your oil glands, and they secrete by rupturing their cells, so we call your sebaceous glands holocrine glands. I'm going to come back to that slide. So most all of your glands secrete this way by exocytosis, but your sebaceous glands secrete this way. They're holocrine glands. So here's another way to structurally classify your exocrine glands. You can structurally classify your exocrine glands this way. I do not want you to worry about this. Please don't spend much time worrying about this at all, maybe just a minute or so. You can structurally classify your exocrine glands by looking at how many ducts they have. If you're a simple exocrine gland, you have one duct leading out. Here's one duct leading out in tan. So here's the secretory unit, the part of the gland that's actually making the substance that's going to be secreted. And there's one duct leading out. Down here, there's more than one duct. Here's 1, 2, 3. So we're not a simple exocrine gland down here. We're called a compound exocrine gland. Down here we have more. All of these have more than one duct leading out. So they're compound exocrine glands. So you're either simple or you're compound. Another way to structurally classify the exocrine glands is the shape of the secretory unit. If you're like this, tube-like, you're tubular. If you're like this, grape-like, you're alveolar. So are you tube-like, or you're alveolar, or you're both? If you're both, you're tubular alveolar? And also, are you branched? You can be branched or not. So please do not spend any time worrying about this. You're either simple or compound, or you're alveolar, or you're tubular, alveolar. But I'm not going to ask you for examples of either of these. Too much information. Way too much information. 1 minute. If you've looked at this, you're good. Okay, that's it for our epithelial tissue. Now, you are going to have quite a bit of information to look over in lab. So we'll spend a lot of time in lab.